The manipulation of genes to create a designer baby is a widely talked about and highly controversial topic. Scientists first began the successful manipulation of DNA in the early 1970s. Before we can begin to apply the possibilities of baby gene manipulation to present-day life, we must first explore the methods of how this may be accomplished. From my research, I have found three major modern-day techniques that scientists are using to attempt to make this possibility a future reality. They are mitochondrial transfer, the CRISPR system, and the CRISPR system using stem cell research. The vast majority of a cell's DNA is housed in its nucleus. However, a tiny portion of DNA is contained in the mitochondria. The mitochondria are organelles that process energy. Many women carry defects that can get passed on to their child through their mitochondrial DNA. This is where the technique of mitochondrial transfer began. The idea is that a donor mother can provide her mitochondrial DNA in place of the defective mitochondrial DNA of another mother. The goal is to then reduce the chances of defects being passed on to a baby. The downside to this, however, is that the child's genetic makeup will be that of 3 different parents, or 2 mothers and 1 father. While there is some hope this technique, it is also a topic of much controversy. According to an article written in Live Science titled Designing Life, Should Babies Be Genetically Engineered?, Lord Robert Winston, a fertility expert at Imperial College in London, argued at a debate that as a result of mitochondrial transfer, abnormal children have been born. He believes in preventing one genetic disease, we are likely to cause another. The second method is germline engineering. It is arguably the most controversial method. It involves the alteration of the genes in an egg, sperm, or very early stage embryo. As a result of this, all traits altered are inheritable to the child and all offspring for all generations. The first type of germline engineering is the CRISPR system. It was co-discovered by a biologist at the University of California, Berkeley, named Jennifer Doudra. This method has yet to be attempted to create a human baby, but it is being attempted in a colony of marmoset monkeys at MIT's McGovern Institute for Brain Research. The CRISPR system is widely regarded by scientists as a fairly easy scientific approach. The approach is used by inserting into a cell a gene-snipping enzyme and a guide molecule. The goal is to essentially attack the DNA letters A, G, C, and T with the objective of altering a specific defective genome. It is most commonly used in the monkeys in an attempt to alter certain genes known to cause childhood autism. Antonio Regaldo, in an article for MIT Technology Review titled Engineering the Perfect Baby, states that CRISPR is currently far from perfect, and that laboratory results yield roughly 20 monkey embryos needing being edited just to get one live successful birth. These are not very good odds to attempt in humans just yet. The second and final type of germline engineering is the CRISPR method using stem cell research. This seems to be the most promising of the two. While embryos cannot be grown and reproduced using the first method, stem cells can. With the ability to edit specific genes within stem cells, scientists can then create an offspring after producing an egg or a sperm. This technique has still yet to make it out of the laboratory, but studies have already produced what scientists call viable eggs. The possibilities of advances in disease prevention and choosing certain physical traits for babies in the near future could be possible using the stem cell approach, given the ability to produce large amounts of stem cells. David Sinclair, a Harvard-based expert on stem cells, states that fully functionable eggs are a when and not an if.